Welcome to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey of Kiss Organics. This is the podcast where we discuss the cutting edge of growing from a science-based perspective and draw on top experts from around the industry to share their wisdom and knowledge. Some quick updates to start the new year. The Cannabis Cultivation and Science Conference is coming back to Bozeman, Montana, August 28th through the 30th, 2026. After a successful first year, we wanted to do it again, but even better. You can see the Collective Elevation facility, the largest in Montana, on the latest Cannacribs episode, and then come and see it for yourself and hang out with many of the podcast guests as well as some amazing science-minded growers. I've already confirmed Suzanne Wainwright Evans and Bryant Mason for workshops and talks. Tickets are now available on the Kiss Organics website. This week's podcast is all about entomopathogenic fungi. It's quite a mouthful, but really just refers to fungi that attack insects. You've probably heard of them even if you didn't recognize the word. Beauveria and Metarhizium are two good examples of commercially available entomopathic fungi. My guest this week is someone with a ton of experience with these fungi and how to incorporate them into your grow. By better understanding their life cycles and methods of attack, you can better control insects or set up a good preventative IPM program. My guest this week is June Graesch. Julie has 18 years of experience as a biological scientist, practiced in laboratory, greenhouse, and field research, as well as technical service support for the integrated pest management industry. She has been with BioWorks for 5 years and has previously held positions at BASF, Becker Underwood, and Iowa State University. Now on to the show. Hey, Julie, welcome to the show.
Hey, Tad. Thank you for having me.
Yeah, I'm really excited to have you back on. I know you are very good friends with Suzanne Wainwright Evans, and you've been on the podcast before, and I'm really grateful to get a chance to talk to you again. I know we have a couple big topics to talk about, the first being entomopathic fungi, which is a mouthful. Can you explain to listeners a little bit about what these microorganisms are and how they work?
Absolutely. So entomopathogenic fungus. Entomopathogenic simply means insect killing. Entom being bug, pathogenic meaning it kills something. So to make it easier, oftentimes we just say EPF. Last time I was on the podcast, we talked about EPN, which is entomopathogenic nematodes. So kind of a similar thing there, but instead of nematodes, we're talking fungus. So how this works is just like many of the other fungus out there. This one is specific to killing insects. And so the sort of the life cycle that's going on there is either the insect comes into contact with a spore naturally occurring in the environment. These EPF are found all over the world, so they're very ubiquitous in the environment, very common, and they are responsible sometimes for creating these epizootics, which just means a mass casualty event in the insect community, although that's typically when their insect populations are getting high in number, and nature often will correct itself. So that doesn't often happen in the naturally in the environment, but we sometimes do see it, even though we aren't applying those products. So they either come into contact with a spore in the environment, whether that is on a leaf or on the soil, or a grower will purchase the product and apply it. So either way, we'll just go off of you're going to purchase an entomopathogenic fungus product and you're going to apply it. So the insects will come into contact with those spores either through primary direct contact, and so that's during the actual application where you're contacting the insect, or you can also have secondary contact, which is the insect wasn't there at the time of the application, but they're coming after let's say a thrips, and they land on a tissue, a plant tissue, or the soil that has been treated with the EPF, and they're walking around and they pick up those spores and they become infected. So, how that infection process occurs is that once the spore, the conidia spore, comes into contact with the cuticle, the insect's skin, it will adhere, so it attaches. And then once the attachment occurs, it will germinate. Germination takes anywhere from uh, 24, a lot of the process that I'm going to go over is going to be occurring within 24 to 48 hours once that adhesion occurs on the insect cuticle. So it germinates. Then it will create a hyphae that will go out and find a spot where it perceives a good location to create a structure called the apissorium. And the apissorium is sort of a flattened section of hyphae that both produces enzymes and pressure on the insect's cuticle to punch through the cuticle, and then the hyphae will go down and get down to the insect's hemolymph or blood. And when it gets there, it does something very interesting and unique. It creates a structure called a blastospore. And blastospores are just hyphal bodies or small fragments of hyphae. And the benefit for the fungus doing that is blood is pumped throughout the body. So if you have a single fungus getting in, the blood then carries those hyphal bodies throughout the insect relatively quickly, causing, the insect to become very sick very quickly and die. In addition, these blastospores are also creating some toxins, and that are going to also contribute to septicemia and death of the insect. So once the insect dies, the fungus will germinate once again. Not germinate, well, yeah, sort of germinate from the blastospore and create hyphae and can then leave the insect host. And this is where you would see potentially a mycost or a fuzzy bug, and you can get sporulation from that. Now, it's important to note here that sporulation often occurs under very humid conditions. So, whether it's naturally occurring or it is fungus that you have applied, you may or may not see that sporulation. You may just see dead insects, and that is normal unless you have very, very high humid conditions. You're more likely to potentially see that on the soil because your soil is typically going to be a little bit more humid than on the plant. So sporulation isn't a function of the efficacy. It's just something that will occasionally happen under the right conditions.
So this process, it sounds like science fiction. In fact, I believe I saw a show called, I think it's called The Last of Us, where they created the storyline around cordyceps in humans. Now, the ones that we're talking about specifically, well, why don't we start there? So, are any of the entomopathic fungi that we would use in agriculture that we might commercially purchase particularly dangerous to humans?
No. So when an EPF is commercialized, if it is going to be commercialized as a biopesticide, it has to go through the EPA, the Environmental Protection Agency. So in order to have a product registered at the EPA, it has to be there has to be a lot of research that goes into it that shows that it is efficacious against the pest that you have on the label, as well as a lot of toxicology that is on the environment, to non-target organisms, to vertebrates. And so you also have to have this toxicology package. And so uh, is in terms of safety to mammals, this is specific. These EPFs are specific to insects. So you can get you've likely come into contact with spores all the time in the environment. Nothing happens unless you would potentially have like an allergic reaction to potentially something that you come into contact with in the environment. It is a fungus. It is a spore. So, in terms of that, you want to make sure that you adhere to the personal protective equipment, the PPE, that is listed on the label for your protection, to ensure that you aren't basically just going to come into contact with those spores and have a reaction in terms of an allergic reaction. But as far as infection, it will not infect. In fact, I think there is a limit at which it goes inactive or is killed, and I want to say that's in the 86 to 90 degrees. Once it's above that, it uh, again becomes inactive or is killed by those hotter temperatures.
And since our body temperatures are higher than that, you're saying it's not a great host for
Exactly,
these insects. Okay.
exactly. We can either consume it, we can get it, in us, and it would just die.
Yeah. Now, some EPF, that's a hard abbreviation for me to remember. EPF can produce mycotoxins. Isn't that correct?
So
But the
that was
ones.
the. Go ahead. Sorry.
Oh no, I was saying the ones specific to agriculture or to horticulture are not necessarily the ones that would do that.
So as I mentioned, the blastospores in the fungus's life cycle, those blastospores are creating toxins, mycotoxins. Myco just meaning fungus. Inside the insect, and that is as an aid to kill the insect. However, once the insect is dead and uh, the life cycle is complete for that fungus, any mycotoxins that escape that insect aren't able to survive by themselves. It's not a it's the mycotoxin isn't a live organism, but it won't persist in the environment uh, once that insect is basically degraded and there's nothing left of that insect cadaver.
So, what are the EPFs that are found in commercial products currently on the market? What are the names of the fungi that you would find?
Ah, that's a great question. So cordyceps, as you had mentioned, is actually one of the products that you can get on the market. You may better know this as Iceria, but Iceria has recently been renamed as a cordyceps. It's not the brain infecting, human one, but it is a cordyceps. So there are a number of products that can be found on the market. Typically, there are three of the most common ones that you find are a Beauveria, Metarhizium, and the cordyceps or the Iceria. Now, these products, there are in terms of the number of products in each one of those, there's probably the most products in Beauveria, and secondly, the cordyceps, and third, Metarhizium. So, and there's also a few key differences oftentimes with these products. So, the Beauveria and the Metarhizium products are typically going to be sold to you as a conidia spore. So that's the aerial conidia spore that is the end of the life cycle. Um, Whereas cordyceps is often, if not always, I hate saying always, but to my knowledge, all of the cordyceps Iceria products are sold as blastospores. So there are a couple of differences with wanting, using one versus the other. There are benefits and drawbacks to each one of those. Blastospores are often long-lived. They have, you'll have a longer shelf life for products that have conidia spore. Um, and the conidia spore will often take a little bit longer to germinate. As in contrast, a blastospore product like the cordyceps Iceria, those are going to germinate a little bit faster because they're sort of an intermediate place in the life cycle of the fungus. So uh, it will often have a shorter shelf life as a product, but a benefit is that it will usually germinate a little bit faster. So you can have faster kill of your insects, and I'm talking, under ideal circumstances, 24 hours potentially difference. So not significantly, but when you're wanting to take down a high population of insects in the growing operation that you're working in, you want to kill those very quickly. What I will say about these various products is that each one of them, each one of the Beauverias, the Metarhizium, the Cordyceps, have their strengths and weaknesses. They're with the various Beauveria products and strains. There are some that are better at killing some insects than other insects. And so, what I always recommend is try a few different products and see what works best in your operation against the pest that you're dealing with. Because in addition to, all these various products, these strains can also have differences in environmental tolerance. So some of them may be better in warmer conditions versus drier conditions. Maybe better. It was potentially isolated from a whitefly, so maybe that one's really good at killing whiteflies, but maybe not as good at killing beetles, for instance. So the best thing for you as a grower to do when you're using these or you are a long time user or a new user of them is I would try several and have a couple always on hand and rotate them because again, they're going to have strengths and weaknesses, and you'll get a bigger benefit, I think, from using them together, not as in the same tank mix, but in a program than potentially just using one of them, unless you have really good results against the pest that you're dealing with in your environment, in which case you could get by with just using a single entomopathogenic fungus. But sometimes it just takes trial and error in figuring out which one works best in your operation.
So I'm, in my head, I've always pictured it like Indiana Jones with these pathologists going out into rainforests and finding a insect that's being consumed by a particular, uh, strain of Bavaria or Asaria or Metarhizium. What's the reality of this? Because I think you do bring up a point that is really important and I want to highlight, which is when we say the word Bavaria, we kind of clump all Bavaria together. But what you're saying here is that they're not all the same. So just to be more specific, let's talk about the product that you've been working on for years, which is Principle, that's relatively recently released to the market. Where is that sourced from specifically? How does that process look to develop a biopesticide?
So that's a great question. And so when you see a label from a registered product. And we can also get into the registered versus the non-registered products, which I think is also a really good topic. But when you look at a product that has been registered with the EPA, so you have pests on the label, um, you'll see at the very top on the front page, you'll see the genus species of that fungus. So Beauveria bassiana in the case of principal. But then what you'll see immediately after the genus species is either some letters or some words behind that, and that is the strain. So the strain in terms of principal is a strain called BW149. BW is Bioworks, so this is one that we have isolated. So in terms of commercializing a new product, uh, you would have a researcher, or you would you have a grower that didn't apply an entomopathogenic fungus, and they go out and they see a bunch of dead insects and they're mycosed, so they're all fuzzy, and they're like, oh, well, what's this? So whether that's, however it's found, at that point you want to culture it. If you wanted to know what that was and you wanted to potentially take it to commercialization, you would isolate it. So you would take some of the spores that was produced from that dead insect, and you would put it on a petri dish plate, and you would grow it. And now you have a pure colony of that. So the first thing you would need to do is make sure that it's an insect killing fungus, or if it died from some other reason, and then you have an adventitious fungus that was eating it after its death. So if you take that spore now that you just isolated on a petri plate and you put it on a few bugs and they don't die, well, then that fungus is just an adventitious fungus that's consuming that after death. However, if it does kill it, then you potentially have an EPF. At which time you would want to probably sequence it and find out exactly what it is, and that would then tell you what the genus species of that EPF is. And assuming that you've now done a lot of research, which would entail what is the number of species that that the fungus will kill, and at what rates do we need, how many spores do we need to apply in order to kill the insect, not just in the lab, but also in the field. So sometimes when we look at things in the lab, they work great, but when we take it to the field, they work not so great, because you have very defined environmental conditions in the lab. When you take them out there, all bets are off on the environment. So let's say you take it out and you make applications in a greenhouse or in a field type situation, and you still see good kill from the fungus that you've applied. So okay, now you have something here. So in order to then commercialize it, obviously, from our conversation before, you would need to run toxicity testing for EPA registration. You would also need to formulate it. So because you can't just apply spores. They're very tiny. They are very hydrophobic, which means that they repel water. So if you just took a bunch of spores, and this would go probably for most spores out there, so not just EPF, you put them on water and they float. So you need to formulate it in order to make it ease of use for growers. So there are going to be several inert ingredients, which is going to help those spores go into solution easily. You could there's usually adjuvants that go into those. There are two types of formulations that you often see with three. You see a liquid formulation, and those are always oil based, because your oils need to be very, very low in water content because water is going to make a fungus germinate. So oils are typically will have very, very low amounts of water in them. And so it's a good media for putting spores into. And the other formulation that you'll often see is a wettable powder. And the third one is a granule. Now, granules kind of have come and gone from the market. I think uh, there is the Met 52 Lalgard M52 product has had a granule product. I don't know if it's still on the market. It sort of comes on and off. Depending on availability, potentially time of year, and then I think also there's a Bavaria Bassiana, the um,
Bioceres.
Bioceres has a granule formulation that sometimes is available on the market. Those can be really great for soil incorporation. If you are mixing your own soil and you have the ability to put a granule product in the soil, fantastic landmines in the soil profile. So those are great products for applying to the soil.
That's
You can also
a
apply
slower
wettable powder. Go
release.
ahead.
Sorry, that's a slower release option essentially. Just that's
It
the main reason.
so um, typically the granule is going to be uh, the media that was used for growing that fungus in production. And sometimes that's rice, sometimes that's a mixture of different things, sometimes it's millet. So there's a whole bunch of different things that Bavaria can be grown on. Now, those types, those seeds are always sterilized. So let's say it was done on rice, and you are applying a granule that is rice. It's not going to start growing in your operation. It's been killed. So that's an important distinction. But so since that is a food source for the fungus, when you apply it, when you apply like a wettable powder or an oil-based EPF, uh, those are just spores in the environment, and they are going to immediately be exposed to things like UV and temperature and humidity, which can cause them to germinate. And if there's no food source there and there's no insect to kill, they would just die. So there's a lot of spores that immediately after application will start to die off for a variety of reasons. However, when you apply it to the soil and you have it be, which is its natural environment, and you have this food source, you can have the Bavaria living on that for a period of time. And so, slow release, I think, is. It's not like a fertilizer slow release, but it can cause the fungus to live longer, uh, um, be there longer in the soil because of that food source that it will continue to consume and potentially sporulate. Those spores are released into the soil as when you are applying applications of water or fertilizer to your soil. Any spores that are produced will then disseminate into the soil and kill insects that you have in there in the soil profile. Um, so those are the three formulations. It is going to depend on the situation on which formulation you want to use. So if you're doing soil incorporation, you'll want the granule. You could also do a wettable powder. You could also do an oil, but you want to be careful with oils sometimes in the soil, especially depending on rates. Sometimes roots can become sensitive to oil-based products at higher concentrations. So typically, when applied to the foliage, you're applying either the wettable powder or the oil-based formulation. Both of them are fantastic products. However, with the oil-based formulation, you do have the addition of suffocation of that oil product. So sometimes you can have a little bit increased efficacy because you get some efficacy from the inert ingredients, in which case, in this case, would be the oil. Sometimes you can also have a little bit more rain fastness with the oil. So if you're applying outside, sometimes the oil-based formulations are more advantageous outside because of that rain fastness. But you could also apply a wettable powder outdoors, in which case I would definitely recommend using something like a wetting agent or an adjuvant that helps that spread. Again, because these products are often hydrophobic, even with some of those inert ingredients that help them get into solution, you still want an even distribution of spores out in the area that you're applying it to. So sometimes by adding a wetting agent or an adjuvant like capsule or silhouette, there's a number of them out there, but you do want to check compatibility, because sometimes when you tank mix these live organisms with another product, it can have a negative effect on the live organism. There's been a lot of compatibility testing out there. Oftentimes, the companies that sell EPF will have those compatibility documents. There are also a few documents out there that are like IOBC and IPM Impact that are paid for services, but there is compatibility information out there. So you want to make sure that those wetting agents are compatible, and then you apply it. You get better coverage with those adjuvants. But it's important with most of the biopesticides, and especially for the entomopathogenic fungus, coverage is key. In fact, I would go as far to say that the biggest failure of biopesticides is poor coverage. The majority of the insects that you are targeting on the foliage are going to be typically on the underside of that leaf. So if you do not have the equipment that is getting good coverage on the underside of the leaf, you're probably not going to get control of that insect, because you have not put the spore in contact with that insect. And let's say with aphids, they are not moving around that much. They plant themselves and they stay there. Whereas a thrips, it's moving around a lot, so it's going to be picking up spores. So coverage is imperative in controlling any insect population.
So, just to kind of go back a little bit on this, basically, there's this huge, long process to bringing a product to market, and all of these products. And so, let's just stick with Bavaria here, where if we talk about the big three in my head, and if I miss one, correct me, would be Principal, the one that you guys just came out with, Botanigard, and Bioceres, in terms of EPA registered, legitimate, commercially viable products. But there's differences.
Also Velifer.
Thank you, Velifer. And there are differences between these products, which I want to highlight as well. So your company, Bioworks specifically with Principal, went through this entire process looking for a strain of Bavaria that would survive in a wide range of host conditions, had efficacy, high mortality rates across a number of target pests, and was also, I assume, relatively easy to culture. And then from there, once you found that, this this silver bullet or this special strain, you then had to create a formulation that would allow for the best coverage on the leaf surface when we go to apply it as well. So it's complicated, and all of these companies are doing this independently, and so you can end up with different results, which you already highlight in the sense that maybe, and I'm making this up, but maybe Principal does better in hot weather versus Bioceres, or maybe Botanigard is better in high humidity versus, another kind of a situation. So we have different host conditions or efficacy amongst different commercially produced products. Is that a reasonable thing to say?
Yes.
All right. So I think that's important for people to keep in mind, because I would just think Beauveria is Bavaria, and it's not. So you do need to do a little bit of extra research around not just the strain itself, but also the method at which you're going to be applying the product based on what your targeted goal is in that situation. One other question I had about, let's just stick with Principal, since that's your company and your product. Where was that particular strain sourced from? What's the origin story of Principal in regards to where it came from?
So that's the so there are sometimes things that are proprietary with the companies. So sometimes you do know where the fungus was originally isolated from. So, like the Apopka, cordyceps was from a whitefly. I think the Bioceres was from a tarnished plant bug. Um, uh, Bavaria bassiana strain GHA. GHA is grasshopper A. So they found a bunch of grasshoppers that were dead, and they were A, B, C, D, so on. And the most virulent one of the more virulent ones, as well as a virulent one that was able to be cultured, which you brought up, and is a very good point. So just because it is excellent at killing bugs doesn't mean that it can be mass cultured. So there are GHA actually wasn't the most efficacious strain, but it was the most efficacious that was able to be mass produced. So getting back to the question of where how BW149 was cultured in our laboratory, but it's not. It's proprietary. What at this point, the insect species that we used to get that from.
Okay, fair enough. When I guess where I was going with this in my head is, I recently talked to someone who said they had a Bavaria that was cultured from a plant instead of from an insect. And so I was curious. So I have a two-part question. One, does it matter in terms of where that original Bavaria came from? And then my second question is, are these entomopathic fungi endophytic with plants? Do they have any systemic properties? And then I guess a third question, while I'm spouting questions at you, would be, are there biostimulant properties or other plant benefits associated with these fungi beyond just the fact that they're going to target pests and insects? Are they going to maybe help increase, I don't know, disease resistance, or offer increased nitrogen uptake, or some of these other things that we use, mycorrhizal fungi for, or trichoderma, or some of these other biologicals? So I just threw three pretty deep questions at you. Where do you want to start?
So the first question was, does it matter where it came from?
Yes.
The second one was, Remind me.
Yeah, are these things systemic or
Ah,
endophytic? Do they infect inside of the plant, like mycorrhizal fungi, specifically endomycorrhizal fungi, the type that we would use with, fruit trees, cannabis, vegetable crops? That endo means it infects inside of the
yes.
root tissue.
Perfect.
And endophytes, like you said, are really popular right now. So, is Bavaria or some of these biopesticides endophytic? And if so, what does that look like?
And
And then the last one
yep.
was, what other sorts of benefits might these entomopathic fungi be giving us beyond just killing insects? Do they offer
Perfect.
biostimulant properties?
Perfect. Thank you. So, does it matter where it came from? So, sometimes it does, sometimes it doesn't. So, most, uh, if not all, of the entomopathogenic fungus do have a pretty wide host range, and range of which in which it can be applied successfully. So, and probably are going to be killing more things than just what is listed on the label. Okay, so they may be better at killing some things than others, and some of them may be better endophytes than others. So it does matter. Uh, However, often, as far as the grower is concerned, really the biggest thing that matters is the efficacy, right? And the result that the grower wants, which typically with EPF is getting control of a pest population. So this to get to the endophytic, which again is a hot topic right now. There's been a lot of recent research on endophytism, and the I would say, a long story short, we'll still get to the long story, but the long story short is that we're still learning a lot about how these EPF, which ones are endophytic. Uh, um, Oftentimes, all of them are, but it depends on the plant. It depends on potentially environmental conditions. It depends on other microbes that are on the plant that it might compete with. So there's a number of things that will, even though it is an endophyte or can be an endophyte, may or may not become endophytic. So there's a lot of things that we know, and there are a lot of things that we don't know. So for me, the jury is still out. I want to see more research, but I am a researcher at heart. And I think there are still a lot of unanswered questions. So what? Let's get a little bit back to, I think, some basics. What is an endophyte? What does that mean? So as you mentioned, endo means inside, and phyte means plant. So inside plant. So you can apply to control an insect population, and it can kill that insect population. But it can also germinate and potentially go into the plant material and live there, and which means that it now has become an endophyte. So if a pest were to come and chew on that plant or suck from that plant, they a couple of different things may happen. If it chews on the plant, it's going to a caterpillar. It's going to ingest some of the Beauveria, and you could have outright death from that insect. But more commonly, you get things like reduced fecundity, which means that it's just less fit. It may not taste good to that insect, and so it may drop off the plant and look for other food that's not infected with an endophyte. You could have still feeding, but less feeding, because there is no other food source, so it's going to stay, but it's not going to be as healthy. It's going to be smaller. It's going to have a longer development time. So you're still affecting the population, um, whether you're killing that insect or not. So sometimes you get insect death. Sometimes you just get reduced fecundity of those, healthiness of those pests that are feeding on it. There are also a number of other things, and there actually was a recent paper by out of Colorado State University, Nia Panwar and Adriana, I don't know how to say her last name, Sczepanic, uh, uh, which is at Colorado State University. And she published a paper June of 2024 on endophytic entomopathogenic fungus as biological control agents of insect pests. And this is a paper published out of the, let's see, and what is this? Entomological science. So, and I think it is open access because I was able to get it this morning without paying for it. So I highly recommend looking at that paper, and there's a really good graphic there. So you have the plant, let's say either naturally occurring or applied, it goes endophytic. The other thing that can happen to the plant is induced systemic resistance, which the plant is turning on pathways that are going to tell the plant, hey, I'm being damaged by this pest. And the plant can have some of its own defenses in killing those insects. So sometimes those reduced fecundity things will happen from the plant and the induced systemic resistance of that plant from the fungus causing the plant to turn on those pathways. Sometimes those metabolites are also going to be found within the plant, and those can be consumed by the pests that will cause, again, reduced fecundity or death of those insects on, insect fitness and survival. Another really interesting thing is that you can also have recruitment from natural enemies. So when you have induced systemic resistance, in addition to helping kill an insect, the plant is also giving off volatile organic compounds into the air as well as potentially into the soil. And that is a signal that, hey, I need help. And it can bring in ladybird beetles or entomopathogenic nematodes to help kill that insect on that plant, which I think is really, really interesting stuff. And that, again, can happen both above and below ground with the plant recruiting help. So can all of these various commercial strains go endophytic? Yes. However, sometimes they'll go endophytic for a short period of time and then they disappear from the plant. Sometimes they will be an endophyte in one plant, but another plant is not a good host for it to become endophytic. So it does seem to depend on the plant species. It depends on sometimes how long that fungus will stay endophytic. As far as you also mentioned the word systemic. So Not in the sense of a true chemical systemic where it gets sucked up through the roots and goes everywhere in the plant. You may find it only in the leaves, and it may not travel away from there, or maybe just the stems, or maybe just the roots.
Hmm.
But it typically does not move. If you applied it to the root system, it may travel up the stem a little bit, but typically it generally stays in uh, an isolated area where the Beauveria, Metarhizium, or Cordyceps came into contact with that initial spore. So there are a lot of things um, about endophytism with all of these different strains. And yes, you can get benefit from it, from pest control. Do I think at this point that it can cause significant control of a pest population? That to me is a little bit up in the air. I think there's potential. But at this point, from my perspective, the more efficacious thing to do would be to make regular applications of the fungus to control the growing pest populations. The conidia spore directly contacting or indirectly contacting is going to cause quicker death of the insect population than the induced systemic resistance or endophytic properties of that fungus.
Now, we could also in the future possibly discover a particular strain of Bavaria that is very highly endophytic and spreads within the leaf tissue and has very good efficacy at increasing mortality rates of insects that are biting that particular plant. So maybe I'm just saying hypothetically in the future, we could discover on a pepper plant, let's say, a particular coevolution or symbiotic relationship between a strain of Bavaria and this and pepper plant that could allow for very, very good efficacy. The potential is there based on what you're sharing, but maybe we haven't discovered it yet, or maybe the research hasn't shown us that. So what you're saying is right now the best way from a insect control perspective would be to do an application of one of these commercial products specifically for the purpose of knocking down the population versus hoping for this endophytic relationship to occur and control it. I think that's really interesting. I was not aware of this idea of VOCs released by the plant being able to attract natural enemies or beneficial insects. That is fascinating. I look forward
Really
to
cool.
learning more about that as research comes along, because I think we're still learning a lot more about plant signaling and how all these relationships happen. It just speaks to the complexity and our lack of knowledge around plant microbe, plant insect interactions. That's so cool.
Yes. And the third question that you had, we kind of touched on it, which was the biostimulant properties.
Yes.
Uh, when you are turning on these various pathways in the plant for the induced systemic resistance, um, you can also cause that plant to have things like increased um, rooting or more root hairs. Again, it's going to depend plant to plant on what that particular plant chooses to do in order to mitigate the damage that is being caused by a particular pest or pathogen. But you can have things like improved bioavailability of nutrients because the plant is putting out more roots, which coincidentally will also pick up more water and nutrients from the soil. So, yes, they can have biostimulant properties as well, but I don't think that has been as well studied either. And so it does get a little bit to the question of that I mentioned earlier, EPA registered versus not EPA registered, because there are some products out there that are either considered a biostimulant. It's probably not labeled as a biostimulant, it's probably labeled as a soil inoculant. And you'll have a very low number of spores in that product. And uh, it would probably is going to have some statement on going as an endophyte, and that you can have these properties season long from a single application. So I would just be cautious of that for a couple of reasons. So we affectionately call these no, so the ones that have been registered as soil inoculants is one thing, but then there's also a number of products on the market that are coming from like India and China, and they say Beauveria bassiana or Metarhizium on them, and they're much less expensive than the EPA registered ones that you can purchase in, the United States or other countries that are registered with the Pesticide Bureau. Now, it's important to note here that anything that is not a registered pesticide, which a biopesticide is a pesticide, right? It's just one that's biologically driven, but it's still registered with the EPA. If you see a product like a Beauveria, Metarhizium, Cordyceps, or even some of the biofungicides, Trichoderma bacillus, they cannot make claims. So they can't say things like, this Beauveria will kill beetles, because it's not registered. The other benefit that you get from a EPA registered product is that it um, uh, has to go through rigorous testing to ensure that it is effective, and also quality control. So the quality control of the products that are EPA registered, that is a Beauveria bassiana strain, whatever. But the products that you get from these other countries that are not registered, they may be completely dead. They may be a human pathogen because they died and something else took over. So you have to be very, very careful if you choose to use a not registered product, that you're not just doing it because of cost. Because, yes, they are less expensive, but they're probably not going to do what you expect them to do. So it's just a general statement of
Yeah.
be
No,
careful
I think
with the
I think those are
unregistered
really good
products.
points. I have a story of from Suzanne, the bug lady, who's we all are familiar with. She went and visited a grower, and she had this 50-gallon tub in the corner. I think they actually sent some samples off to Dr. Michael Brownbridge to analyze, because they claimed it was Metarhizium. And it was really interesting because at the time Metarhizium was unavailable due to a supplier or registration issue. I can't remember which. So people were desperate for it, dealing with brood aphids. And so they got this tub in from, I think, from India or China. And basically, long story short, they found it didn't contain Metarhizium, or it was very low levels. I can't remember which. It did have some human pathogens in there. You're also risking potentially heavy metals in there if it hasn't been tested for that, as well as they might just put in some kind of nasty actual chemical pesticide and that they claim is one thing and it's totally another. So yeah, people do need to be very careful with that. And I've heard the sales pitches around like, why they're not EPA registered, because it's expensive or it's taking a long time. And yes, it will kill these things. We just can't put it on the label. Those are all things that you will hear from people pitching you these non-EPA registered products. But because they aren't registered, they're not regulated. Because they're not regulated, you don't really know what you're getting. So I think that's a really important point. I wanted to get back to a question that popped up in my head around this idea of endophytes still. So I guess the question is, are there any negative effects to endophytes or to that infection occurring by these commercial products that are currently available? Because when I think of, and I know plants are not humans, so I want to state this, I'm just using this as an analogy. If I'm doing something that is going to create an immune response in my body, high white blood cell count, that tells me that I have an infection occurring, my body is putting energy into creating those white blood cells, into creating that immune response, which I assume would limit my output or performance down the road. So is that the same with plants, in the sense that if it's having to allocate energy towards an immune response, this systemic acquired resistance or whatever it is that going to limit my potential yield or its ability to put energy into other aspects of plant growth?
That is a fantastic question. So there can be. Again, I think this is an area in which is being studied. In the paper that I had mentioned earlier, there is a challenges section, so I recommend reading through that fully. One of the things that they mention in that paper is a negative interaction with natural enemies, especially natural enemies that may feed from the plant themselves. They may be taking nectar or pollen, or maybe not pollen, but sometimes they will suck juices from the plant or the extra floral nectaries, in which case you can cause potentially a negative interaction with a natural enemy. Also, if a natural enemy potentially feeds on something that is fed on a plant that had an endophytic entomopathogenic fungus, that insect is not as healthy. And so you could still have some negative impact on the natural enemy from feeding on an insect that is somewhat impacted or even infected. There can also be plant drawbacks, as you mentioned. So if you are turning on these pathways that were not on before, you can very much divert, instead of, say, what I talked about earlier with um, stimulation of root development, you could have potentially smaller plants, because it has diverted energy from growing to plant defense. Sometimes those are fairly minor. If you were doing a side-by-side trial, uh, you can sometimes see little differences in height. So is it enough to say, I'm not sure I want to use that because I don't want, slightly smaller plants? Um, uh, I don't, again, I don't know if there's enough information out there truly um, on entomopathy and the impacts on yield to point to at least to my knowledge, point to a literature piece to say yes it can, or no it doesn't. But we do know that it can impact it. So I think it's important to be aware of it, but I think the risk is fairly minor. You may see an effect, but it's not probably going to be significantly noticeable.
Okay, yeah, I've never heard of a grower, because growers use these products all the time. I've never heard of a grower come back to me and say, I got lower yields from applying Beauveria or Iceria or Metarhizium, but it is something, I mean, a lot of times I find if you're having to apply like these products, specifically in my head, Metarhizium, you're probably dealing with a root aphid outbreak that's severely impacting the health of your plant. So anything you can do to boost plant health or to fight down populations of these insects is going to make a huge difference in overall yield and plant health. So that's typically how I see these products being used, where you already have a problem that's having a direct impact on yield and plant health. I did have one final question that I thought of that kind of goes back earlier into the podcast to kind of wrap everything up. But one of the reasons that I know Suzanne loves and promotes beneficial insects, these natural enemies as we call them, is that pests cannot develop a resistance to being eaten, right? Unlike these pesticides, traditional chemistries that we use, pests can, if ones that survive can evolve resistance to that chemical or that pesticide, and then future applications, let's say pyrethrins, for example, wouldn't be as effective, which is why we rotate pesticides, rotate chemistries, and things like that. With entomopathic fungi, can insects develop resistance to the fungi itself? Have you seen that? Or is this sort of in the category of beneficial insects, natural enemies, where they just they can't really defend themselves against it?
That's a fantastic question. So there is, if you're not familiar, there is a website called IRAC. There's also FRAC, and that is the Insect Resistance Management Committee. I think there's also a lot of really good information at Michigan State University on resistance and where we're seeing resistance in various products and compounds. There has been no cases of resistance to any of the entomopathogenic fungus. Now, I think as you would with something that is sort of your lock and key, where you have a receptor and it either is going to fit with that receptor and kill it, or it's had a mutation and it can no longer fit into that and cause death, in a very simplistic way of thinking about resistance. When we're talking about entomopathogenic fungus and also a lot of these other biopesticides, they have complex modes of action. It's not just one thing that's going on, it's a lot of things that are going on. So in terms of the fungus, I think a really big component of what's going on there, that I had mentioned in the life cycle, is when we get to the blastospore and those little fungal segments, which really resemble a little bit more of a yeast. So insects have immune responses, and those immune responses can prevent infection of insects by causing melatonin to, encapsulate and so on. And so that still goes on with a fungal infection. And initially, its immune response will be triggered. And oh, this is a fungus. And then all of a sudden it changes and it seems, oh, wait a second, is this a yeast or a bacteria? And so it confuses the immune system as to what this organism is. And then all of a sudden it's creating these mycotoxins that are also contributing to the death of that insect. And so we don't see resistance development with things like entomopathogenic fungus because of that really complex life cycle. Now, what you might there may be behavioral adaptations that could affect it being infected. Oftentimes, bugs can sense with antennae and feet that something is there. Even there's something that I am sensing that might kill me, and they go away. So that's an avoidance behavior. So there may be some behavioral things that insects may do. but not in terms of what we typically think of as insect resistance. Maybe really, really thick cuticles can have an effect on the fungus's ability to puncture the cuticle. So I think there are certain adaptations that insects have evolved over a very long period of time to have. Um, and we sometimes study those, and we don't see very high control with some insects. Aphids is a good example. Aphids are often on entomopathogenic fungus labels, uh, but sometimes we don't see as good control with it. And I think the reason for that is, say, they don't move around, so they're not secondarily picking them up. And under ideal circumstances, which greenhouse growing is typically going to be ideal environmental circumstances, aphids can shed their skin every day. So you apply a fungus to an aphid that's not an adult, that's not going to shed its skin. An adult's not going to shed its skin anymore, but the juveniles, the nymphs will. And so if they shed their skin every day, then those spores just get shed off. And it takes 24 to 48 hours typically to get to the point of cuticle penetration and blastospore generation. So oftentimes you need to pair an entomopathogenic fungus with something like an insect growth regulator, like an azadirachtin. Actually, Suzanne and I looked up in Iraq and the MSU database for azadirachtin resistance. And we did not see any cases with azadirachtin because, again, it has this complex mode of action. And azadirachtin is a botanical bioinsecticide. So by pairing an insect growth regulator, you slow down the insect's life cycle, you allow the fungus time to go through that first couple of stages within its life cycle, and then you can kill it. So I think there are certain things that you can do to increase the mortality of insects that are harder to kill for entomopathogenic fungus. Sometimes you need higher rates to kill certain insects. So it really kind of depends, which is why it's important to look at the label, make sure you use the proper rates. And one other thing that I have learned through working with growers for a long time with entomopathogenic fungus is that it's oftentimes better to make low rate applications frequently than one or two really high rate applications infrequently. So it's a numbers game oftentimes with fungus, and and the number of spores will gradually and sometimes quickly decrease. And so put a few spores out, and then in a week, 7 to 14 days, put more out in small little bursts. So you're always kind of keeping that spore level up so that when you have more insects that are generated, especially ones that generate very quickly and have really fast life cycles, you're going to need to do frequent applications. And you don't need to do high rates if you're doing frequent applications. So that's one of, I think, the biggest, if you take nothing else away from this, if you're using entomopathogenic fungus, tank mixing is your friend as long as there's compatibility, and making frequent applications at low rates is a good strategy. And adjuvants if you're using the wettable powder formulations.
Well, that was fascinating. I learned a ton in this podcast, so I really appreciate it. I love, I love having you as a resource, and I'm very grateful that I got to know you and befriend you through Suzanne and her network, amazing network of hyper intelligent people that are so incredibly knowledgeable on these very specific things that are so interesting. So thank you again for your time today. We didn't even get to some of the other topics that we wanted to talk about, so we'll absolutely have to do this again.
Love to.
Yeah, thank you for coming on.
Thank you very much, Tad. I appreciate it, and it's always a pleasure.